Well, first I would like to share my experience while asking my colleagues this question, asking at conferences, meetings, in a personal communication, do you apply PET or molecular imaging to study brain connectivity? What or how? It's at least a half of answers. No, a quarter of answers. Do you mean fMRI? A very painful answer for me. And yes, 5%. Now I cannot answer you this question. I cannot ask you this question, but I will reformulate it. And I would ask you to raise your hand if you are familiar with analysis of fMRI data. Who is familiar? Okay, great. Analysis of PET data. Okay. Analysis of fMRI data in the context of brain connectivity. And analysis of PET data in the context of brain connectivity. Okay, a few ones. Good to have an impression. Outline, I will briefly mention brain connectivity as a dominant concept of brain function. I will compare PET and fMRI. I think this comparison is very instructive. Then I will briefly say what is molecular connectivity and how can it be estimated. I will then come back to the comparison and now compare molecular versus functional connectivity. Then I briefly introduce the molecular connectivity working group and give an overview or outlook for the future. So first, I would like to say or postulate that PET is an established neuroimaging method. How can one say if the method is established? In my eyes, the method is established if it has a regular practical utility. And here are a few examples of clinical indications where PET is routinely used in clinical settings. So one example is neural function in dementia and movement disorders using FDG PET, neurotransmission using tracers such as FDOPA and plumazanil in movement disorders and epilepsy. Protheinopathies is an increasingly important topic. There are multiple amyloid and tau tracers in the meanwhile, which I applied to diagnose patients with suspected dementia and movement disorders. And finally, amino acid transport can be studied using amino acid tracers in brain tumors. So in the cognitive neuroscience, in the last 20 years, there has been a change in the paradigm. Specifically, the idea that a particular brain function is governed by a single brain region has been replaced by the concept where brain functions rely on a coordinated interaction between several brain regions, which are often referred to as networks. Here you see an example of 6 so-called resting state networks as identified in fMRI data. And there is a steadily increasing interest in brain connectivity. Here is a PubMed search for brain connectivity. You see 40 hits in 1990, while there are nearly 5,000 hits in 2020. So if brain connectivity would be a stock, and you would invest in brain connectivity in 1990, 10,000 euro, now you would have been a millionaire. But in my opinion, it is still not too late to invest in brain connectivity or in brain connectivity research. and I will explain why. Here is a beautiful figure by Ariana Sala, who performed a PubMed search and combined terms brain and connectivity and a method, neurophysiological method. So there are a number of methods which could be separated in the functional domain. So these are mostly functional methods like near-infrared spectroscopy, MEG, EEG, PET, and fMRI. And there is a sort of structural domain with structural MRI and diffusion MRI. And we can easily see that around 3/4 of functional domain studies are done using fMRI. And I would conclude so far that the current perspective on how brain connectivity and networks underpin brain function disproportionately relies on the hemodynamic signal of fMRI. And I hope that fMRI experts would not strangle me at the Faculty of Law, but I would also say that there is an obvious discrepancy between the huge number of studies, fMRI studies, and little practical use. And I would like to continue with a comparison between PET and fMRI. This is quite arbitrary comparison, so don't be too strict, please. If we compare, let's say, average spatial resolution between FDG PET and Bolt fMRI, by the way, why I would like to compare these modalities, because both measure or both are markers of neural function, and FDG PET is by far the most popular tracer FDG, while Bolt fMRI, as we know, is by far the most popular method in the field of brain connectivity. Temporal resolution is of minutes in FDG PET, in seconds in Bolt fMRI. There is a star. Why? Because there is increasing interest in so-called functional PET. You have heard probably of research by Sharna Jamada, where FDG is injected not as a bolus, but continuously. And this allows potentially to increase temporal resolution up to a few seconds. Data mode, typically static versus dynamic. Scan duration is a bit longer for PET as compared to Bolt fMRI, and radiation exposure is present in PET, but not in fMRI. And costs, obviously, also very important difference, are much higher for PET than for Bolt fMRI. And I would say that this data acquisition mode, radiation exposure, and low costs explain, at least to a substantial part, the popularity of fMRI as compared to PET. But FDG PET is closer to real neural activity, so it measured in a more direct way, I would say, as compared to Bolt fMRI, where we deal with a surrogate of cerebral blood flow, cerebral blood volume, and cerebral rate of oxygen metabolism. So it's a less direct measure, and signal-to-noise ratio is higher for PET than for Bolt fMRI. Reproducibility is known to be higher again for FDG PET. And as a consequence, there is no wonder that FDG PET has a wide clinical utility, while it is not the case for Bolt fMRI. And as Daniela Pirani explained, obviously it's not only FDG PET, and there are multiple targets which PET can approach. And here is a beautiful overview, again by Ariana. So some important targets have been already approached by PET or by molecular imaging, but some not. There is much research in this area. So let's say with PET we have a large repertoire of potential targets. And given all these arguments, I would say that molecular imaging at least deserves to be evaluated in the field of brain connectivity, even if so far the proportion of PET studies has been much, much lower than the proportion of fMRI studies. What is molecular connectivity? Molecular imaging of brain connectivity. I would define it as a statistical dependence between regional measures of molecular imaging. It's a wide definition, yes, and it echoes the definition by Carl Friston, who defined functional connectivity as statistical dependencies among remote neurophysiological events. How can molecular connectivity be estimated? Well, there are two principal ways: intersubject and intrasubject. Intersubject, as I mentioned before, this is, let's say, mostly governed by functional PET studies, where fluctuation in FDG signal can be studied within one subject. It is quite a new approach. And intersubject, so most of the literature by far on molecular connectivity is based on intersubject estimation. And this is, I think, what many people are a bit afraid of. So how can you estimate molecular connectivity when you have just one single image per subject? It is different for bolt fMRI, obviously, where you have fluctuations of the bolt signal, and you assume that two regions are connected if these fluctuations are synchronous between two or more regions. So then you assume a functional connectivity. In PET, we have just one image per subject, and we perform analysis across multiple subjects. And this arises a very important question in biology, medicine, and beyond about ergodicity. So how equivalent are patterns of intersubject covariance to patterns of intrasubject covariance? What is ergodicity? It is interchangeability between variability across time and across ensembles. General definition. And I think ergodicity is underestimated, or let's say the importance of this phenomenon is underestimated in medicine. If you think of, for example, clinical trials, right? We assume that a certain intervention works if there is a decrease in a particular biomarker at the group level. There is a significant difference in one group as compared to placebo group. And you assume, or let's say the conclusion is that the drug or intervention is efficient. But it is the group level. Can this conclusion be true for single subjects? Obviously not. But so far, again, this phenomenon hasn't got much attention. This phenomenon is old and has been discussed in many fields beyond medicine and biology, and I found this discussion very nice by Anthony McIntosh in the course about analysis and function of large-scale brain networks already in 2010. who wrote that some scientists are concerned about which source of variance across tasks. So he is talking about functional MRI. Across tasks or across subjects is best for estimating neural interactions. The issue of which source of variability is correct is not unique to neuroscience, and there is no necessity for making a logical connection between covariances computed across tasks within subjects and those computed across subjects within tasks. However, there is also no justification for preferring one source of variability to another, particularly in cases where both can be examined, as in fMRI or event-related potential studies. Within subject analysis assesses the direct relation between regions, while across-subject analysis indicates the stability of that relation. They are complementary and not contradictory pieces of information. And for me, this is a good justification of this inter-subject estimation. And I would like to present you a few examples where this ergodicity can be or has been shown. So fMRI data, functional connectivity, as you know, both fMRI data can be downsampled in dimension down to one single map per subject. Yes, this map is called fractional amplitude of low frequency fluctuations. So basically, it shows you in amplitude of the Bolt signal across region. And if you perform inter-subject analysis, so you perform covariance across subjects using just one single map per subject, like in PET, right? So it's the same for PET. You come up with very similar so-called resting state networks if you calculate them in a conventional way from averaging inter-subject Bolt fluctuations. So this is a conventional way, Bolt, and these are maps from amplitude of low frequency fluctuations. And you see that they are very similar. I would say at least there is a certain degree of ergodicity. It is not black and white. Of course, it is a continuum, but at least some degree of ergodicity in functional connectivity can be assumed. And this is a nice study by XCND, not yet published, who I didn't even know that, but in the ANI database, there have been subjects who have been scanned 5 to 9 times. And he calculated or quantified an association between cross-individual and within individual variance. These are the results. So the regional matrices for different regions of interest, but essential is this plot with R value of 0.77. So there is a strong correlation between within subject and between subject variability. So I think this data also prove that there is at least a substantial degree of ergodicity in PET data, in FDG PET data. And yeah, in my opinion, this inter-subject data may be suitable to characterize an invariant population-based portion of the connectome. If you are interested in this topic, or let's say in the methodological background, I would recommend you these articles. Okay, now let's move to a comparison between molecular and functional connectivity. Yeah, it's a rather old study where we compared in the same subjects. These are the same 54 subjects who have been scanned simultaneously in the PET-MR. So fMRI data and PET data were acquired simultaneously in the same subjects, and we see concentration of variance, cumulative, and we see the number of principal components you see that the first 5 components, principal components, explain around 30% of variance in the fMRI data, while the first 5 components explain nearly 70% of the variance in FDG PET data. It means that there is a higher variance concentration in FDG PET data as compared to Bolt fMRI, which is typically translated into a higher statistical robustness of PET data. And this is very important for replication. So if you find a difference in one sample and you want to replicate it in another sample, your perspective, your chances to do this are much higher for PET than for fMRI. Here is a study by Aldana Lizaraga, a PhD student of mine, who calculated reproducibility, test, retest, so 2 sessions, again, FDG PET and Bolt fMRI data. So you see here Pearson correlation coefficients, functional connectivity, and this so-called FDG covariance across subjects. We defined and called them yes connections, all connections with a PCC above 0.5. This number is much lower in the fMRI data. And in the second session, we recalculated the analysis. Basically this line is most important, which shows the proportion of connections in functional connectivity and FDG covariance data. And we see that with FDG PET, we can, let's say, cover a larger proportion of the brain as compared to functional connectivity in a reproducible or, test-retest in a reproducible way. Recently published study where we analyzed in how far structural connectivity, so we applied diffusion MRI to reconstruct neural fiber tracts connecting brain regions, and we calculated an overlap between the structural connectivity and we call them functional or proxy measures of functional connectivity, to which belong functional connectivity itself, then covariance of FDG signal and covariance of gray matter volume. And we see that this is functional connectivity, so around 50% of functional connections have a structural substrate. And this follows by FDG covariance, so very close to each other. So we conclude that structural connectivity also underlines at least around half of FDG covariance connections, similar to functional connectivity studies. And this is not self-understood, because some people argue, well, you calculate covariance across subjects. This is an artifact. This is not a real biological signal. And if this would be so, I would expect that FDG covariance would be closer to gray matter volume covariance, because they are calculated in the same way, right? We have also T1 image, 1 image per subject in structural MRI data, and 1 image per subject in PET data, but this is not the case. So they are, let's say, the functional measures of brain connectivity are much closer to each other. And final slide on this comparison, work done by Isabelle Rip, a former PhD student of mine, who compared clinical utility of PET and Bolt fMRI data. So we used ICA to define some, let's say, established rest and state networks. We can call them also cognitive rest and state networks, like posterior, anterior DMN, default mode network, salience network, central executive network, and we use two rest and state networks as reference, which are supposed to be not related to cognition, primary visual and auditory network, and we this were also simultaneously acquired data for three groups: patients with Alzheimer's disease, with behavioral variant of frontotemporal dementia, and healthy controls. And as you see, in the fMRI data, there was a sort of general decrease in functional connectivity across all rest and state networks. So not limited to cognitive networks, only the posterior default mode network demonstrated a difference between healthy controls and patients. But in the FDG-PET data, this pattern was much more specific. There was no difference between the reference rest and state networks, but there were differences between the cognitive rest and state networks, and in a way that we can not only differentiate between patients and healthy subjects, but can also differentiate between Alzheimer's disease and behavioral variant frontotemporal dementia. And recently, we have established an interdisciplinary team of experts from different fields who wish to, let's say, to evaluate molecular imaging as a tool to study brain connectivity. The team has become larger in the last times. There are also younger scientists joining the group, and we are happy also to have Matej on board. And our major tasks are, which is at the same time also an outlook, is the validation. We want to understand what we actually measure, what is the meaning of this covariance, or let's say molecular connectivity. We want to very carefully understand and validate methodology, establish so-called best methodological practices. So how reproducible, how replicable are data? Simulation studies are very important. How dependent are these covariance patterns on different experimental conditions, for example, number of subjects or acquisition technique. So the second aspect is application. We are building an atlas, at least now for FDG-PET data, of molecular imaging maps of the human connectome based on data with established PET traces. We would like to integrate molecular connectivity into the framework of causal models to enable causal inferences on brain function. And we want to derive and validate this molecular connectivity indices at the individual level. Ariana Sala will give a talk now on this aspect, and the colleagues in Ljubljana has been very successful on this applications aspect. So to use this molecular connectivity in the differential diagnosis of neurodegenerative disorders. Very great, important work. And finally, we want to build resources for the community to enable people who don't work with PET data or don't have PET data actually, but are interested in this molecular connectivity, so that they become access to PET data, they become tools, and get an overview of studies on relevant topics. Yeah. And this is a sort of perspective. So obviously, there is no one connectome. It's black-white, right? Or it's color? on the slide. On the original slide, it's colored. But yeah, I see it like white. It doesn't matter. So you see here so-called fingerprints, structural fingerprints based on tractography, so reconstruction of fibers, neural tracts, then functional fingerprints from EEG, MEG, and other techniques. Here, PET, glucose metabolism, PET dopamine synthesis, and also, proteinopathies. The idea is, I think it is very important to integrate this connectivity information, and this would facilitate understanding of the brain organization. Yeah, I wish you, in the end, to have fun with molecular connectivity, as I do, and I thank you for your attention.